Gene activation by a growth factor is a process in which a growth factor causes a chain of events which leads to the mRNA being synthesized and sent out of the nucleus to give specific instructions to the cell. In the beginning of gene activation by a growth factor bonds to a receptor protein through a process called diffusion. After diffusion, the receptor protein becomes active. The active receptor protein activates a relay protein, which then activates protein kinase 1, as shown here. When protein kinase 1 becomes active, it produces an ATP. The ATP then gives up a phosphate group, turning itself from ATP to ADP, and the phosphate group latches onto protein kinase 2, making it active. The protein kinase 2 produces an ATP, which then gives up a phosphate group, turning itself from ATP to ADP, and the phosphate group latches onto protein kinase 3, making it active. The active protein kinase 3 produces an ATP. After this happens, enzymes called protein phosphatase remove the phosphate group from protein kinase 2 and 3, making them inactive. The ATP produced travels into the nucleus where it activates a transcription factor. Once the transcription factor is activated, it reads a part of the DNA where the growth control genes are found. From this, mRNA is synthesized and is sent out of the nucleus to give instruction to the cell, and that is the end of gene activation by a growth factor.